Hi, I'm Phil Basford. Today I'm going to be talking to you about our sample and data management system for micro-CT based X-ray histology. As I'm sure many of you have experienced, data management in a research lab can be a major challenge. In our case, this is particularly so for two reasons. The first is that we are a high throughput lab. We have a sample autoloader that will enable batch scans to be run overnight, producing large volumes of data for us to process the next day, and each sample may be scanned in multiple different ways and in multiple different states. We have two Nikon micro-CT scanners, but we also have a Dectris detector which will enable us to do photon counting based scans. However, the biggest challenge is the fact that each sample may be scanned multiple times. A typical workflow might be a tissue sample arrives and is scanned fresh. It is then frozen and scanned again. It is then formalin fixed and paraffin embedded, scanned again. Finally, it is sectioned and then each slide can be processed with different stains and imaged under the light microscopes we have access to. All this data and all the different processing steps applied to the sample need to be recorded. To do this, we have developed a management system which records this data, provides automatically generated previews of scans to enable us to find the relevant scans, and assists with automatically generating reports that can be handed over to the user. For more information, please see our website.
Welcome to my presentation on the low count reconstruction for hyperspectral or colour X-ray CT. So we call it colour because we use an energy sensitive detector, so we can measure the position but also the energy of every incident photon. So that extra information can help us identify elements within our sample based on their spectral properties. So our first aim was looking at the image quality improvement we get for low versus high count datasets. We did this using open source software known as the Core Imaging Library. So we've got a phantom sample here as an example. You can see that when we use the FDK method, for low counts, we actually get a lot of noise and poor feature definition as you'd expect. But if we instead use iterative methods, and in particular we apply what's called regularization parameters, we can apply noise reduction and edge preservation that allows us to get image quality similar to or better than much longer scan dataset. Secondly, we wanted to highlight the spectral analysis that's possible with the improved reconstruction, and we did this with an iodine-stained lizard head. So now you can see the same smoothing and noise reduction, but this time in the energy domain, and in particular in this region which we use to identify the signal as iodine from this K-edge. If we isolate this signal, what we can do is K-edge subtraction and segment out the iodine-stained soft tissue from the remaining skull structure. This is some of the stuff we can do with hyperspectral CT, and it leads to a lot of exciting future possibilities, particularly in the area of multiple staining of biological specimens.
Hi, my name is Jakob Larsson, and I will talk about 4D imaging using a liquid metal jet X-ray system. Time-resolved X-ray CT captures dynamic processes and has applications within the medical field as well as in food and material sciences, for example in situ imaging. Conventional lab-based systems have difficulties in operating simultaneously with both a small X-ray spot and short exposure times. Our phase contrast system is based on metal jet microfocusors and enables fast, high-resolution imaging of centimeter-sized samples. As an example, the video shows air bubbles inside a dough while rising. It is composed of 7 tomographies, each with a total acquisition time of 57 seconds. With further optimizations, the acquisition time can be reduced to below 10 seconds per tomography, primarily by faster camera readout. This would enable even faster time-resolved studies in a laboratory environment. I should also mention that the technology behind this work is in the spin-off company of Exiscope. Thank you for your attention.
My name is William Tvengström, and I will present phase contrast imaging of cancer tumor tissue samples. In cancer surgery, assessing the resection margin is very important. Today, the standard method is histology, based on rigorous sample preparation and light microscopy. Contrast and resolution are excellent, but the method is destructive. It only gives two-dimensional images, and the sample preparation takes about a week. A fast three-dimensional method to image resection margins potentially has several patient benefits, including improved patient outlook. We propose phase contrast CT as a method that fulfills the needs. phase contrast gives much better soft tissue contrast than conventional micro-CT. The resolution is sufficient to see cellular-sized features in 3 dimensions, and it's much faster than histology. The images here were taken in 3 hours, and we are aiming for 20 minutes. Since it doesn't require any contrast agents or other sample preparation, the time from when you get the sample until you have results is considerably faster than the 1-week histology. Since phase contrast micro-CT is non-destructive, you can still do histology afterwards. I shall also mention that the technology behind this work is in the spin-off company Exiscope. Thank you for your attention.
Cochlear implants are neuroprostheses which restore hearing by replacing the function of damaged hair cells inside the inner ear. Despite the great success of cochlear implants, some individuals do not achieve the anticipated benefits. One of the reasons for this could be the immune response to the electrode array. We have established a mouse model of cochlear implantation to investigate the biological response at the electrode tissue interface and to investigate whether changing the material that coats the array affects the immune response. Micro-CT imaging has been used to visualize the mouse cochlear in 3D pre-implantation and to visualize the position of the array post-implantation in order to validate surgical technique and also to identify any soft tissue damage. This is a whole skull scan where the cochlea and implanted array have been highlighted. These images on the right are 3D volume renders where the implant and bone have been segmented using a VISO in order to identify the position of the array inside the cochlea. The overall aim of my project is to investigate the biological response to cochlear implantation, to ask what is happening at a cellular and molecular level and how does this influence cochlear implant outcomes. After imaging, I'll be processing, sectioning and staining the tissue to corroborate the micro-CT and histological data to provide novel insight into this biological response. Thank you for listening.
Hi, I'm Kirsty Alsop and this lightning talk is on statistical evaluation of forensic tool marks using micro-CT. Specifically, I'm studying saw and knife marks on human bones and predicting information on the tool used to create them. This is forensically relevant for dismemberment cases and homicides involving knife crime, where current evaluation methodologies involve subjective human comparison of surface striations. In my project, experimentally created tool marks are needed. To be representative of forensic casework, human cadavers must be used. Tool marks are visualized using micro-CT and 2-dimensional cross-sections of each tool mark are extracted. Micro-CT is a great technology for this technique as the tool mark is digitally preserved and cross-sections can be taken non-destructively. 7 measurements from each cross-section are then used to develop random forest prediction models for a more objective tool mark evaluation method. Regression random forest models have successfully been built to predict the thickness of saw blades from the measurement data. This is shown to accurately predict the saw blade thicknesses of saws within the model more than 95% of the time. Classification random forest models will be built in the future to predict saw features and hence narrow down potential suspect weapons in forensic cases. Applicability of the technique to knife marks will also be studied. The models will be validated using past police cases to establish the applicability of the methodology to real-world casework.
Good afternoon, I am Roxane Bonito from the University of Portsmouth. Critical side bone defects remain a surgical challenge for which the considered solutions must combine optimal biological and mechanical properties. And in this context, biodegradable magnesium fibers are promising candidates due to their mechanical properties close to trabecular-borne ones. In this study, we focused on the biological aspect, employing XCT to image and compare the bone formation induced by the magnesium fibers with 2 different controls, an empty defect and commercially available calcium phosphate particles implanted into dog mandibles. At 16 weeks of implantation, the magnesium fibers have induced more bone regeneration than the other, both visually with the restoration of the continuity of the defect and quantitatively with a significant higher regenerated bone volume fraction. This is probably due to the smaller size of the fiber and faster degradation with the release of magnesium ions that promotes closer interaction with the newly formed bone. Additionally, the presence of both osteocyte lacuna and blood vessels cavities suggests the bone undergoes physiological remodeling and angiogenesis. I like to believe now you have a better picture of the ability of the magnesium fibers to promote osteointegration, conduction, and induction, allowing the restoration of the critical size defect. and thank you for your attention.
Hello, and welcome
to Jurassic Park.
No. My name is Robin White, and today I'll be discussing applications in correlative multi-scale lab-based tomography. I'm going to assume you're all familiar with the two systems, the Zeiss Versa, which allows for non-destructive high-resolution imaging at the macro scale, and the Zeiss Ultra, which brings us truly to the nanoscale, and instead highlight the main objectives in performing correlative imaging. These are to inspect and navigate large volumes for precise nanotomography, reveal complementary information across length scales, and target specific features for extended destructive analysis such as TEM. Here's an example where we use this workflow to observe specific attachment points of an apodeme, shown in blue, from a biological suction cup after locating the Versa. This allowed for the apodeme to be tracked and targeted in the Ultra and imaged non-destructively, giving researchers the ability to understand exactly where and how it connects to the control suction cup function at the V-notch. In another view, we can see the stark difference in resolution and the clear visualization of fan fibers and connections to better understand the larvae's attachment mechanism. Here we see how this can be beneficial in modeling and in energy materials, shown here through a porous network system where higher resolution resolves the missed pores. In addition, we can use machine learning to upscale the Versa by 10 times to give a more representative structure used in diffusion modeling over a large volume. And with that, I'd like to thank you for listening.
What if cartilaginous fishes had bone? This ambitious question was raised 40 years ago and put on standby since then, although it conditions our understanding of the evolution of vertebrates. The standard state for skeletal elements of sharks and rays is cartilage covered by small mineralized blocks called tesserae, but a bone-like tissue has been identified in the vertebral neural arcs of 2 sharks and 1 skate. This bone-like tissue has been cautiously renamed lamellar mineralization and is morphologically characterized by the affixing of layers of mineralized matrix surrounding elongated cells. Micro-CT images make the distinction of such patterns especially useful even though the cells are not visible. We sampled the neural arcs of 20 species among 11 of the 16 extant groups of chondrichthyans and combined histology and micro-CT approaches to identify lamellar patterns. The advantage of micro-CT images is twofold, providing a fast, reliable clue of the mineralized matrix morphology and emancipating from histology biases due to the limited number and hazardous orientation of section planes. Our results reveal that lamellar mineralization is widespread in sharks, skates, and rays, suggesting an ancestral lamellar condition that persists in more groups than expected so far. A fine comparison between this tissue and bone is still necessary to say if sharks and rays really have bones.
Welcome to this presentation about kitchen-based light tomography. We have developed a home-based scanner in order to perform tomography in a home environment. The first scanner was developed in the kitchen and therefore the name KBLT. In the new generation, we're using a Raspberry Pi and a touchscreen and a graphical user interface to control the rotation stage, the web camera, and the light source. We have looked into both transparent and non-transparent samples, and we have also set up workflows to perform the reconstruction using TomoPi. We have also made a reference scan using normal X-rays, and we have also set up workflows to perform the image processing and image analysis work. And finally, also the 3D renderings. We have also looked into samples which are transparent but have some internal defects which we're able to reconstruct and render. And following the evolution of tomography, we have also looked into samples which change over time, for example the sand in this hourglass, and we have also used so-called sample environments where we can force the samples to undergo changes. And KBLT will allow you to develop your physics skills, your electronics, IT and math skills, and finally also your scientific skills. And the skills that you obtain from working with KBLT are directly transferable to normal X-ray neutron tomography experiments. And with that, I thank you for your time.